Thanks, Laura. Thanks, Matt. Welcome, everybody, those who are to the stage, and everybody else who have been patiently listening. Just to remind you before I open the floor for questions, in case you don't want to be recorded, you can ask to mute or something. Right. With that said, welcome to this, and if possible, keep it as brief as possible so we can go through as many questions from the audience as possible. Thanks, Andrew.
Thanks. Could you tell us a little bit about the regulatory process that occurs for animals? I'm sure it's not easy. Is it difficult, let's say, as the FDA, or is it a smooth process? Thanks.
Sure. So I can tell you what my experience has been. I will say I'm perhaps not the best person to answer this when it comes to the process with FDA. So FDA in the United States regulates veterinary medicine and human medicine, and you can absolutely go after a veterinary, an FDA approval for veterinary indications, right? And that process, this is my intuition and my little bit of understanding, is still pretty complicated, although maybe not quite as complicated as it is for human clinical trials. But it is a real sort of deeply bureaucratic process that you have to go through if you want to get FDA approval for a veterinary medicine. In our case, because we're an academic project based out of academic institutions, the regulations that we are subject to are institutional animal care processes. To the extent that we collect human data, the IRB, although we've been designated as not having to get IRB approval, at least so far, and then the mandated rules. So for the clinical trial in particular, right, all of the procedures for the Dog Aging Project undergo institutional animal care approval at the relevant institutions. For the clinical trial, we also have a data safety monitoring board, just like you would for a human clinical trial. And I will say that we actually, as the Dog Aging Project, have made it part of our culture to go above and beyond when it comes to animal safety and ethics. And in addition to the Data Safety Monitoring Board, we also have an Animal Welfare Advisory Board that is not mandated, but that we put in place to have extra oversight over everything that happens at the Dog Aging Project. So those are the kinds of processes that we have put in place for the clinical trial for Triad. We are not seeking FDA approval for rapamycin in our study for lifespan. So we have not gone through the steps of trying to get the study approved by FDA with the goal of then getting the drug approved for as an aging drug in dogs. Part of the reason for that is that rapamycin is already, I'm sure as you know, off patent and available as a generic, and veterinarians sometimes, it's not commonly used in veterinary medicine, but it is sometimes used for certain types of cancer and things like that. So this is not, from our perspective, about trying to get FDA approval.
Thank you.
Thank you, Andrew. Go ahead.
Laura, jump in, please, anytime. Thank you, Andrew, and thank you, Matt, again. Aaron, you're next.
Yeah, so the most interesting thing about this project for me is the comparison between the castrated and non-castrated individuals. Can you talk more about that, or your plans for any kind of data you're taking for that?
Sure. Yeah. Great question. Yeah. So first of all, I'll just start by saying that it's not often appreciated until you think about it that there are actually 4 sexes in companion dogs. There are intact male, intact female, sterilized male, sterilized female. Yeah. So it's a really interesting question. How does sterilization affect age-related outcomes, and is it different between males and females? So we definitely capture that information, absolutely, and we will be able to correlate health outcomes during aging with reproductive status as well as biological sex. So that's absolutely something we're looking at. The one thing I will say is, at least in the United States, it is relatively uncommon that companion dogs will be intact. Most dogs are spayed or neutered. Companion dogs are spayed or neutered, sometime early in life. So we are, relative to the sterilized dogs, we're underrepresented for intact dogs, and that's a group that we are trying to recruit actively for. But even still, with 32,000 dogs in the study, we have several thousand intact dogs. And so we will be able to start asking those kinds of questions. I can't tell you right now what the answer is because we're sort of at the beginning, but I agree it's a fascinating question, and absolutely we expect that spay and neuter status or reproductive status will have an impact at least on the spectrum of diseases and disease risk that goes along with aging. There might be small effects on lifespan just based on what's in the literature now, but it doesn't seem to be a major determinant of lifespan at this point, although we'll see what happens. For Triad, we made the decision for Triad that all of the dogs enrolled in Triad would be sterilized. And the reason for that is that by that age group, the proportion of dogs that are intact is so small that we would have been underpowered to detect effects associated with reproductive status. So we just made the decision that we would take that out of the potential complications in the analysis, and all of the dogs are sterilized that are going to be enrolled in Triad.
Okay.
Nice. Thank you, Matt, and thank you, Aaron. Chris, would you like to ask your question?
Hi guys. Yeah, so my question is a bit of a speculative one, and I know how scientists hate to answer speculative questions without about a million caveats, but I'll go for it. So what are 2 or 3 things that you believe are inevitable for the future of longevity over the next 5 or 10 years, and what obstacles may prevent these from happening?
Yeah, good question. So this is a prediction which I'm terrible at. So I'll just start by saying I'm wrong more often than I'm right, but I'll give it a shot. So I think one thing that we'll see for sure are registered clinical trials. I mean, this is not really much of a guess because it's already starting to happen. Registered clinical trials for aging, and my hope is that within the next 10 years, certainly, we'll see the first FDA approval for a geroscience intervention. That probably, if it's in the human space, won't be for aging as an endpoint, but everybody's going to know, right? As these trials get designed in a smart way, I think people are going to know that that's really what's being measured, even if that's not what FDA calls it. So I think that'll happen. I certainly hope, and I think certainly in dogs, there's a good chance that we'll see the first FDA approval for something to directly target biological aging. And there, the endpoint might actually be aging or something like that. I actually think that's going to be a watershed moment in the field once that happens, because then it's proof of principle. That's the way I think about Triad, right? I think, the fact that we've got Triad off the ground is proof of principle that you could do a clinical trial for aging in pet dogs, and now other people are doing the same thing, which is great. And I think that'll be the same with the first FDA approval for a drug that targets aging. So I think that's going to be something that will happen and will be really important. I think we're already seeing the sort of flourishing of small biotechs and industry coming into the field. There have been a couple of false starts over the last 20 years where it looked like that was going to happen, and then some very obvious big failure, and everybody backed off. I think we're over that sort of barrier now. And this is happening. There's a lot of interest in the biotech world in the biology of aging. The field has matured to the point where there are lots of different strategies that can be taken. So I think that will continue, and I think that's going to have a big impact on the field. What do I see as the challenges or potential barriers? I think still we have a real challenge in the field in convincing serious people who control federal funding that the field is ready for investment. This is obvious by the fact that geroscience is still funded as less than 1/2 of 1% of the NIH budget in the United States, despite the fact that biological age is the greatest risk factor for all the diseases that NIH, almost all the diseases that NIH. We have not been successful at convincing the policymakers and the serious people who control where federal funding goes, that this field is to the point mature enough to warrant that kind of investment. I hope that happens in the next decade, but it's going to be hard, and we have challenges with that there still are high-profile individuals, some of whom are scientists, some of whom talk about themselves as scientists, who say crazy things that turn off serious policy people. So I worry about that, and I don't know how long it's going to take. I think it's great to have a donor drop $25 million on the field, but that's a drop in the bucket compared to $2.5 billion, which is what the NIH could move into the field like that if we could convince them that we should do that. So that to me that's where I would like to see us be successful. I would like to see the field be recognized in terms of resource allocation at the federal government level that it deserves. And I think, honestly, you could make a case, and I wrote an op-ed in The Hill a couple of weeks ago, right, that, 6.5 billion is actually small compared to what I think the field deserves in terms of resource allocation. But we've got to convince the people, again, these are serious people, and they don't want to talk about immortality or stuff like that. They want to talk about rigorous real science. And I think that's a challenge that we've got, and it's still a bottleneck in the field.
Amazing.
Thank you, everybody. The questions have been so great so far. Matt, I just wanted to be mindful of your time. You're okay with answering some more questions?
Yep, I'm good for a while longer.
Okay, awesome. Because I think we're going to have some other great questions. And I did want to agree 1000% with you in terms of how we speak about what's happening. I've had conversations with friends in the field, and when we say life extension, immortality, it's not making people, or it's not gaining us fans. Even if that's what people want, it's not what most of society is looking for. But if we can get our dogs to live longer and we can live healthier long, I think people would be more interested. And that's just my soapbox that I get into. So I'm going to let everybody else ask their question now. Angie, thank you so much for being here. Please go ahead and ask your question.
Hi, everybody. I'm very happy I'm on stage and here with you. And I think this pet longevity project is freaking amazing. And I'm happy that I finally hear about it. And I have a quick comment on something, Matt, you mentioned so many different amazing discussions. I'm getting my bio age for the reason that I think I'm, in midlife, but I'm super cool and fit, right? But now if I get this test done and I'm not, my organs are not 20 years old that they are, then that will be a real kick for me, and so I'm doing it for the empowerment of me to say then, oh my gosh, all these things I'm putting into tomorrow, like more running or this or that, I will do now because I really, want to live as long as I like. So anyways, I think that's one of the reasons we might want to do that. And then my dog is a mixed breed, and I just filled out your form, and I wondered if the breed and what the breed means in your studies and what you have observed so far and in your decision process, and also small practical question. I have basically made up the breed of my dog and called her Tibetan Terrier after my veterinarian said that's probably it. And then since I did that, every second person on the street who before said, is she a Shiatsu? They all said, oh my gosh, where's the Tibetan Terrier? So I put that in, and I hope that's good enough. And that's a question that maybe other people are also interested in. How specific do we have to get with the breed of our dog when we fill in your forms? And then the other question was, what breed does the breed impact your process, your choosing, and what have you seen so far? Thank you.
Yeah, great. Thank you for that. Yeah, and I'll say my sort of comments on the epigenetic clock test. Absolutely, if you want to get the test, go get the test, right? And obviously, everything I say comes from my perspective. But certainly, if getting the epigenetic test is going to motivate people to have, healthier lifestyle habits, it's great. The only point I was making, and I think this kind of ties into your breed question as well. The only point I was making is that right now, those kinds of tests, the epigenetic clock or the breed test that you can get for your dog. So you can go, there are several tests that you can get that will tell you the breed of your dog. They're not actionable at this point. There's no, really good sound medical or lifestyle changes that you would make based on those tests for medical reasons. So that's the only point I was making. But I think it's great if it's a motivating factor. So for the dog aging project, first of all, thank you for nominating the dog. That's great. Mixed breed versus purebred. The data seem to be the case that normalized for body size, mixed breed dogs live about a year longer than purebred dogs. That's just observational, but that does suggest that there are potentially real differences, certainly in longevity and maybe in aging. For the Health and Life Experiences Survey, we absolutely recognize that owners are going to do the they can in answering the questions around what breed is your dog, and that oftentimes it's going to be wrong, right? So that's actually an interesting aspect of the Health and Life Experiences survey is all of that data is owner reported. And then we compare that with the medical records, but for things like breed, right, the medical record is not going to be any more reliable necessarily than what the owners think. So we recognize that. There are a few questions in the survey that help us understand the degree of perceived certainty that the owner has around the breed, but we recognize that it's not as good as a DNA test. Does that influence selection into the what we call the sampled cohorts, so foundation, precision, triad? The answer is yes, it does, but not in any way that is important. The way it influences selection is that we try not to be overrepresented for any given purebred breed of dog. So for example, if we didn't do that, we would probably have a ton of golden retrievers. So we limit the number of dogs from any one common breed, but other than that, it doesn't really have an impact on which dogs are invited to participate in the sample cohorts. And there's no limitations on breed for the pack.
Thank you, Matt and Andrew, for the great questions in terms of signing up. We're going to go to Nihil. Welcome to the stage. Please go ask your question.
Hey, hi. Thank you, Matt, for staying up and taking questions. My part of my question was to ask how many dogs are enrolled in this dog aging project, and I think I had it 32,000 in one of your answers for the previous question. So it's really interesting to think about the amount of data that is going to get generated over the years of the progression of this project, because the current my question is more about your opinion on the following two issues when it comes to studying this data in terms of data science and big data analysis. So in general, when we have a data set, we often remove the exceptions and we normalize the data, and that helps us what exactly happened in these exceptions, what has gone wrong or what has gone correct. In such cases, exceptions by the definition mean that they are exceptions and we do not have a lot of data around exceptions. So how does one accurately assess that a certain thing is a causation in case of exceptions? That's the first part of thing. I'm purely talking from big data and analysis on the data that is going to get generated, and an extension of that is the primary endpoint is of course lifespan, and that just assesses how long will the dog live, but we are between two age-related diseases in terms of age. If you do not have a proper disease A progression equals more aging, then disease B progression which equals less aging. So we can
both good questions, both challenging questions, especially challenging to answer in a concise way, but I'll try. So I think with the exceptions, this is going to be a bit unsatisfactory, but it really depends on the specific type of exception or extreme individual, right, that you're talking about. So one way we're approaching this is I didn't mention this, but we do have a small cohort of dogs that we call the centenarian dogs. These are dogs that live exceptionally long for their body size. And so there we're actively going out and trying to find these exceptional agers and sequence their genome and ask what's different about their genetics and their environment. So that's an example where we're actually doing exactly what you said. We're seeking out the unique individuals that might tell us something important about how you achieve exceptional longevity. In other cases, it really depends. So we talked about how a lot of the data we collect is owner reported. And there, the exceptions are often going to be owner mistakes. We don't want to get bogged down by including that data that is, like if an owner says that their 80-pound dog lived to be 37 years, we can be pretty sure, unless they can provide documentation, that that's a mistake. And so we are less likely to include or more likely to throw out those extremes on owner-reported data unless we can actually document the validity of those data points. So that's the way that we're thinking about that kind of data. And honestly, I'm not a statistician, and that's above my pay grade in terms of when it comes to the actual analysis, how do we handle the details of the data. We've got really good statisticians on our team and big data people who are sophisticated and know how to do that. Okay, what was the other question? Sorry, my age-related cognitive decline is showing itself. What was your other question?
The other question was, how do you assess the effect of rapamycin on healthy?
Yeah, and that's a really good question, and that's important in any study of aging, right? Even if you look at the mouse studies in this field, you will see lots of cases where people claim that they have extended health span. So first of all, this is my personal pet peeve. I use the word sometimes incorrectly myself. If anybody tells you that they have shown that intervention X extends health span, they're wrong. They have not done that because we do not have a quantitative way to measure health span, which I think is what you're getting at. There is no quantitative measure of health span that is accepted in the field. Every paper that has the title X increases health span in it is misleading. And I will say that, recognizing completely that I published a paper in 2016 that had that phrase. I've come to change the way I think about this. But the point is that, yeah, we don't have a good way to assess whether an intervention has actually broadly improved health span. I think in people, health span is something where you know it. Does somebody have a good quality of life and good health? But even there, we don't have a great way to assess what's that tipping point where you go from having health span to your health span is ended, right? Not only that, but at the individual level, you can go below and come back above. You could have good health span, something happens to you, you're sick for a while, you drop below the arbitrary health span threshold, and then you come back up and you're healthy. That happens all the time. So I think these are all conceptual challenges that we face when we're trying to assess whether an intervention is really broadly impacting biological aging. I don't have a great answer for you, except to say that the way I approach it is, first of all, I think lifespan is the gold standard. That's true in laboratory studies, and I think it's going to be true in the real world. That's the one quantitative metric that there's no ambiguity about, right? Either an intervention makes individuals live longer or it doesn't compared to the control. So I like lifespan for that reason. But I certainly recognize that it doesn't capture everything that we want to capture when we talk about the aging. And that's why, I try to look as broadly as possible. So I do that in our laboratory studies in my lab, whether that's in C. elegans or in mice. We try to look at multiple functional measures of aging. I'm going to come back to that, and molecular measures, and look as broadly as possible. And in Triad, that's why we have multiple second endpoints. It's for exactly that reason. That's why we're looking at heart function and kidney function, and liver function, brain function, activity, muscle function, for exactly that reason. Now, your specific question of, let's say rapamycin, improves heart function but nothing else, or improves kidney function but nothing else. How do we interpret that? I don't know. I mean, I think we'll get there, and then we'll see what the data look like. My gut feeling is that if rapamycin only impacted one age-related outcome and not the others, at least not multiple of the others, I would interpret that as it didn't broadly slow aging in dogs in that context. But I think we've got to get there and see what the data look like. And do is look at multiple functional measures. And honestly, that's more or less what a frailty index is, to broadly assess health as opposed to only looking at one thing.
Thank you, Matt, and thanks, Nikhil. Alina, you're up next, and the last few questions, so Matt and Lawrence after that. But yeah, Alina, you're up next.
Great, thank you. Hi, Matt. So in humans, there's theories that like stress and traumas could cause, early onset of disease. Maybe they don't know what happened when it was a puppy, that kind of thing.
Yeah, that's a good question. So, I mean, the answer to your question you kind of already alluded to is that we're doing the best we can within the constraints, right, of the population, right? So you're absolutely right that some dogs will come from a shelter, right, in middle age, and the owners won't know much about its history before that. Other dogs the owners may have had since it was a puppy. So we're collecting all of that information, including potential traumatic events. And traumatic events, could be obvious traumatic events, like the dog got hit by a car, but it could also be events in the dog's life, having to move because either the family relocated or there was a divorce, the dog went with one owner and not the other. So we're trying to capture as much of that information as we can. It's going to be correlative at best, and that's also where we're at. I agree with I think what you were suggesting, which is that those kinds of life experiences are likely to be important for health outcomes and potentially for biological aging. And we're doing our best to capture that information, but just given the nature of the data set, it's going to be an imperfect data set for us to actually really tease out a lot of those effects, I think. We will also, of course, get other information that could be also contribute to stress, but is also, likely to contribute to health, such as if it's always lived in an urban environment versus a rural environment versus a suburban environment. What's its typical day like? Does it spend most of its day outside, most of its day inside? How many other dogs are there in the house? All of that stuff is information we collect, which I hope will kind of all go together to paint a picture of, the experience that dog has in its environment, and some of that certainly could contribute to trauma and stress.
Thank you, Matt. And Matt, you're next.
Great job. I had a question about how has it been working with local vets for sort of biospecimen collection and ensuring quality, and has there been any challenges? And second question, is there any post-mortem analysis as part of the dog aging?
Yeah, both good questions. So I think we're still a little bit early on to know exactly what the challenges are going to be with the private practice veterinarians. So far, I think on that end, it's gone pretty smoothly. So one thing I'll say is private practice vets are extremely busy and underpaid individuals. And so we've done everything that we possibly can do to make this as easy for them as it can be, to make it as little work as possible. And most private practice veterinarians are heavily invested in the health of their clients, and they are excited to participate in a project like this. So I think it's been pretty smooth. Honestly, I think the biggest challenge we've run into with sample integrity at this point is I don't know what's going on with FedEx, but they're struggling right now to get stuff delivered overnight. So if I had to point to one thing, I think that's been our challenge with sample integrity. But hopefully that'll get better. I'm dissing on FedEx a little bit if anybody's listening. So, but the vets have been great. Sorry, what was the other? Oh, necropsy or autopsy. Yeah. So we are in fact asking the owners when they nominate their dog if they would be willing at the end of the dog's life to donate the body. And we have actually a biobank at Cornell. So the director of that is Marta Costajano, and she's the director of the Cornell Veterinary Biobank and also the Dog Aging Biobank. And she has experience in facilitating donation of bodies at end of life for dogs and other companion animals. And so the plan is that at least when feasible, we will collect tissues and organs from dogs that have been in the Dog Aging Project, either in the longitudinal study or triad, for those kinds of end-of-life studies. That is going to be particularly important for our studies of dementia, because as you can imagine, Alzheimer's disease is really a disease that's diagnosed at death on autopsy. And so being able to actually look at brains from dogs that developed dementia during aging in normal life will be really important for understanding to what extent does that mirror Alzheimer's disease or other dementias in people. So yeah, that's something that we have incorporated, and it'll really honestly just be a matter of time to see how smoothly that goes. There are a lot of challenges, as you might imagine, to that process of facilitating.
That was perfect to end the question for Matt with another Matt. Thank you so much, Matt, for spending this time with us and for kicking up this series with us. I guess I wanted to, you did say that you are looking for funding for the Dog Aging Project. Can this also be, are you guys taking also collections from private individuals, or are you only looking for grants and bigger donors?
Taking donations from anybody who wants to donate. If you go to the Dog Aging Project website, of course, there's a donate button. You can click on that. People actually should have two options. You can donate through the University of Washington, where I'm at, or through Texas A&M. Sometimes if they're alumni for one or the other, but it's all administered through the universities, all tax deductible, and we would absolutely be grateful for donations from anybody who is so inclined.
That's great. Now everybody knows, and I know that Nathan can probably have some good ideas of very bigger donations. But I just wanted to say thank you again, and thank you to my co-host Avi and everybody who is here. And we hope that you come back the next 4 weeks, as we're going to be talking to other amazing scientists working in the field of pet longevity. Avi, did you want to please? I'd like to say something before the end of
Thanks, Laura, for everyone who is here, and I hope that Matt agrees with this as well. And this show created by Laura is called Let's Talk Pet Longevity. So we are planning to bring on scientists that are working cat longevity as well, and cat as well. So because obviously we care about dogs are awesome, and so are other pets as well. Yeah, we're going to be continuing this series with other scientists on improving the longevity of other animals. Matt, this was absolutely a pleasure to host you, and thank you for doing all the work that you're doing on the dog aging project, and hopefully in the near future, all of us benefit from the work that you and your team. So with that, Matt, I'll let you draw this to a close if you don't mind.
Sure. First of all, it's really been my pleasure to join you. So thank you for putting this on. And thanks to everybody who's listened in. I think you can probably get the idea that I'm pretty enthusiastic about the dog aging project and about the field for geroscience in general. And so I hope that you share that enthusiasm. There's going to be a lot of really exciting developments over the next few years, and I'm energized to see what happens. So thank you all for joining us.
Yes, thank you so much, and a lovely weekend to everyone, and hope to see you next week again. And thank you to the audience and for the great questions. They were very good, took it a step further than anything Avi or I could have done. Matt, thanks again, and a lovely weekend to everyone. Hello?